Okay. All right, great. So let's get started again. So now, after kind of Rudy has introduced some of the ideas about why you might like to use a method like DIA, it's my job to try and give an introduction to what that actually means. And I should say that this material for some people, I think, is going to be revision and introduction, but for the purpose of the first day, I think we need to get people up to the same speed. So if you are one of the more experienced people in the audience, then kind of bear with us. There's more advanced material coming, but it's important to also go through these fundamental parts. So what I'd like to talk about essentially is why do we need DI? Well, this is pretty much covered by Rudy already. I'll just sort of gloss over this. What do we actually mean by data independent acquisition? And what are the kind of principles of operation of the instrument? And also, what are what's the kind of fundamental idea of how we analyze the data? And then we can sort of take a look at what might be some advantages of DIA and maybe a little bit of outlook. So why do we need something other than DDA or SRM? So as I said, Rudy kind of showed a picture something like this already, but let's just remind ourselves that this is probably the proximal goal that we're trying to get to. There's some matrix of samples or conditions and a matrix of peptides or proteins, and what we want to have is this matrix filled in a complete way with high quality quantitative values. And Rudy also showed a slide something like this. So this is kind of a historical motivation. So some people will argue that this is certainly not reflective of the state of kind of DDA analysis today. But let's say 7 or 8 years ago, when we were kind of going in the direction of DIA, or 10 years ago or more, when people were going in the direction of targeted proteomics, there was some kind of dissatisfaction with the type of data matrices that were produced by DDA. This pushed part of the field, at least, in the direction of targeted proteomics, where you could get more kind of complete and reproducible data over many conditions. But for the bottleneck was, of course, the smaller number of proteins. And so just before we get into the DIA part, let's remind ourselves of what the data looks like for DDA and for methods like SRM. And now I'm going to show a bunch of projections in this kind of 3 dimensions that we have always: retention time, mass to charge, and intensity. And if you look at from this projection, what you're going to see is basically a mass spectrum, MZ versus intensity. And if you look in this projection, you're going to see chromatography like ion chromatograms, for example. And I just want to just to make sure everyone's on the same page. We're going to look at what this looks like for DDA and for SRM, and then later on move on to how it looks for DIA. So if we're talking about DDA, this is now MS1 scans from the DDA method, from the DDA method, and what you see is that each one of these gray lines is a slice in time, is a precursor scan, and we have these 3 or 4 peptides eluting, and you can see them eluting chromatographically as a function of time, and you can see the isotopic patterns kind of hiding behind here. And what happens is, well, of course, you can extract ion chromatograms on the MS1 level if you want to do quantification. But for the moment, what we're more interested in is what's happening in the MS2 space. So the instrument is instructed to, on the fly, decide which peptides are going to be sampled, which parts of the precursor space are going to be sampled for MS2. And what you get is snapshots of basically MS2 spectra that correspond to a given point in time in the precursor in the MS1 space. And you get these snapshots, and you can identify these peptides, of course. So the point is, the precursor space is sampled by MS2 spectra discontinuously in both the mass and time dimensions, right? So these little boxes, which represent the precursor isolation for a given MS2 spectrum, are kind of dotted around this map in a kind of stochastic way, or in a semi-directed way, but you don't cover the whole space. This is so this can be very fast. So we can get a high number of identifications in a sort of qualitative sense. It's instrument driven somewhat, so it can be biased towards the high abundant species, because this is what you're telling the instrument to do. The heuristic is on the fly, and it can be potentially inconsistent across samples. In contrast, in SRM, if we look at the same picture, what we have then is now we instruct the instrument to sample the precursor space in a deterministic way. So we say we're interested in this blue and red peptide, and we're just going to ask the machine to only select this precursor space over and over again in a deterministic way. So just go for the red one and the blue one and alternate between these. And what we get is a recording of fragment ion spectra, which is also essentially deterministic. So we've programmed the instrument in advance to know what precursors and what fragments we're interested in. We only get information about those. And the thing to notice here, and of course we can extract quantitative information by integrating these fragment ions as a function of time. And we can project them like this, and you'll see a lot of pictures like this later in the week. But the main point I want to make here is that the precursor space is sampled by MS2 spectra, or MS2 signals, in this case, continuously in the retention time dimension, but it's discontinuous in the mass dimension, right? So we can't, the instruments aren't fast enough to sample the whole MZ precursor space where we would expect peptides to be with these narrow precursor isolation windows. So continuous in time, but discontinuous in the mass dimension in terms of the precursor space. And so this can be consistent and can lead to accurate quantification, and it's entirely user-driven because the machine is forced to select the low-intense signals if you ask it to, but the problem is that we have a low number of precursors monitored per run. So this is kind of a revision now. So let's get on to what we mean by data-independent acquisition. So just a little bit of history. I mean, this term was introduced in 2004 by the Yates lab in this Nature Methods papers. So what do we mean by data-independent acquisition? Well, let's say what we don't mean. We explicitly are not talking about data-dependent, right? So there's no precursor, stochastic precursor selection happening. It's entirely deterministic. So it has this fixed duty cycle, and the instrument just does what you tell it to do without trying to make any decisions by itself. So this is, in a sense, philosophically more like SRM or PRM, but unlike DDA. However, on the other hand, it is also entirely on-target and unbiased data acquisition. So this is, you don't need a target list, you don't need to pre-specify what the peptides you're interested in. So in this sense, it's kind of more like DA, but unlike SRM or PRM. We're usually trying to achieve some kind of comprehensive sampling of the precursor space with MS2 spectra, and this typically is coming, the way that we achieve this is by using wide precursor isolation windows in the most common case. And the implication of this is that we usually end up having very complex mixture MS2 spectra. So we'll say what this means. But this is basically the features, and we can kind of go over this at the end and see whether you agree with that or not. In terms of a historical perspective, this is now a figure from a review now accepted, actually this should say accepted in molecular systems biology, which will come out in the next few weeks, which is kind of a tutorial on how to do SWATH or DIA. And as part of this, Tina, who will also be instructing on the course, made this figure where we have sort of time axis on the Y here. So the earlier methods are at the bottom and the later methods are at the top. And it's also sorted by the precursor isolation window width. So this is this original DIA paper I mentioned from John Yates. There was a paper also from Dave Goodlett's lab at the same time, which tried to do a similar thing, except without a precursor isolation window. They tried to fragment everything. This kind of ended up looking a bit like MSE in the end. We're mostly operating in this space here. So we're talking about SWATH mass spectrometry. And we have this fixed window method here, and the red one is this variable window method. But the point of showing this slide is that there are a very large number of methods published, and they all have kind of different names, and it can be a bit confusing. So I would say that all of these methods fall under the umbrella of data-independent acquisition. And just to make the distinction, you will hear people use the term kind of SWATH and DIA interchangeably, but what we usually mean by SWATH is something operating in this regime. And I just think it's also interesting to look at what the development and application rate is for methods like DA or SWATH. And so this is a PubMed query that tries to capture something about how much this is being used. And what you can see is that basically there was a lot of kind of technical and proof of concept studies happening during the 2000s, but it didn't really take off until about 5 or 6 years ago. And essentially what happened was basically that we got faster instruments, but we also got smarter data analysis. So the algorithms got better and people people thought of better ways to interrogate this kind of highly complex data. And this led to a lot more intense method development, and now I would say by now we have kind of practical implications and implementations and people doing a lot of application-focused studies. And so the number of citations is still relatively small, I would say, to other areas in mass spec-based proteomics, but it certainly seems like it's upward trend. Okay, so let's go back to these projections and see what we're actually talking about when we mean DIA. So here's our 4 peptides again, and these are, as it happens, in the range from 400 to 525. And so now in this SWATH method, what we're going to do is basically open up the precursor isolation window. So again, this red box is depicting the precursor isolation window. We're going to collect deterministically over time. We're going to co-isolate whatever is in this precursor isolation window from 500 to 525. And so this blue one and this red one and the gray guys are all going to be co-isolated, and they're all going to be co-fragmented, and the MS2 spectra that we acquire in the end is going to be a mixture spectra from these different peptides. And so you can see the fragment ions here are kind of colored for clarity, but in the real world case, of course, the ions aren't colored, so what you just have is this very, very complex mass spectra, which are very difficult to interpret. So at any given time, you have this one snapshot of this MS2 spectrum, which contains most of the peaks of any of these given peptides, but also other signals. And actually, if you know something about what these peptides look like from previous analysis, then you can potentially try to selectively extract the signals that you want out. So if you say, I only want to look for these blue or red signals, well, then you might be able to make sense of this very, very complex spectra. But the main point I want to make here is that these MS-MS signals are basically continuous in the mass and time dimensions. So each one of these precursor isolation, wide precursor isolation windows allow you to have sampling of the precursor space continuously, both in the retention time dimension, but if you stack a bunch of these wide precursor isolation windows next to each other in the mass dimension, then you're able to have essentially complete sampling of the precursor space with MS2 spectra, but it comes at the cost of having very, very complex MS2 spectra. So again, the kind of the features of the data is that the instrument is forced to fragment all precursors in every cycle. So what we get again is consistent acquisition of MS2 spectra for everything that's there, right down to the kind of, well, we have to put a caveat here and say within the limit of detection of the instrument. And this should lead to reproducible quantification capabilities, as is the case in SRM type of data. But again, the cost is this very complex data, and we need to figure out clever ways to extract the information. One thing to mention at this point is one constraint that we always need to think about, which we don't necessarily need to think about in DDA, but we do need to think about in SRM, is chromatographic sampling of these peaks, right? So we need to know something in order to we're doing quantification based on extracted ion chromatograms. And this is now a figure from actually an SRM example, but it's what it tries to illustrate here is how the duty cycle of the instrument relates to the chromatographic peak width and whether this is a constraint or not. So you see, if you set the duty cycle to 2 seconds, which is this green and blue lines, you have very nice kind of sampling over the chromatographic peak, and you'll be able to reconstruct this peak well and get accurate quantification. In the case of these longer duty cycles, like 10 or 20 seconds, then we don't get so many data points over the peak, and you can see that this pink line or this red line is not going to do a very good job in terms of being able to reconstruct this peak and to estimate the peak area, which is ultimately what we want to know. So just to keep this in mind. And now I just want to reiterate, so this kind of these 3-dimensional plots some people don't really like, so now I'm going to show the 2-dimensional version of this just to hammer this home. So if this is the whole precursor space, and basically this is now retention time versus M over Z, probably most of you are used to looking at some kind of a figure that looks something like this, and each one of these little black dots is a peptide. And the way that we operate the instrument now is to say, okay, we're going to break up this precursor space from 400 to 1200 M over Z into chunks of 25 M over Z windows. So we need 32 of those to cover the whole space. And each one of these precursor isolation windows is what we call a swath. And so this is basically the ensemble of these swaths repeated over the whole run so the ensemble of the fragmentine spectra acquired throughout the chromatographic range is what we refer to as a swath. And essentially the cycle time, so what I was referring to in the last slide, the space and retention time between these two cycles of the instrument is the thing that we need to take into account, that it should be, we need to be able, if we have a peak width of sort of 30 seconds or something like this, well then we need to have a data point every 2 or 3 seconds in order to reconstruct this peak. And so the spacing in between these little blue arrows is significant, and this is what we refer to as the cycle time. So the time required to come back to the acquisition of the same isolation window. And so if we're going across 400 to 1200, and we want to cover that whole space, and we know we need a cycle time of roughly 3 seconds, and we know that in order to get good quality spectra, we need some kind of minimum time. So at the time we were using about 100 milliseconds, and that means we're allowed to have roughly 32 swath windows in this method, and that means that each window needs to be 25 mass units wide. So there's kind of a balance here in terms of the chromatographic cycle time, how long you want to spend on each MS2 spectrum, and the width. So obviously as we have larger width, precursor isolation window width, we're going to co-isolate more peptides, and the data is going to become more difficult to interpret. But as we make this window width smaller, the data is going to become higher quality and essentially easier to interpret. So just to kind of re-summarize this, because this is basically important, I think, to understand what the data dimensionalities are. In DDA, we have this sampling of the precursor space, which is kind of stochastic and also, or at least semi-stochastic, and is not continuous either in the retention time or the mass dimensions, but rather we get these snapshots of peptides all over the map. That's very useful for doing identification. In SRM, we make the compromise that we're not going to cover the whole mass to charge space, but instead we're going to sample continuously in the retention time dimension, and we're only going to acquire data for the things that we're really interested in. And then DIA is some kind of attempt to make a compromise between both these positions, where we want to have continuous sampling in the retention time dimension, but we also want to have information about as many peptides as possible, so we want to have continuous sampling in the mass dimension. And we get, this allows us to cover the whole precursor space, but it comes at the cost of these very complex mixture spectra, and we'll talk about later on how to try and get the information out of this. And so now this, for those of you that are interested where this kind of swath name came from, this was actually Ludovic Gillet, or in fact his wife, who was a scientist working in this type of area. If you want to make a map of the Earth using satellite mapping, what you do is you fly the satellite around the Earth and you measure kind of strips or swaths of the Earth all at once, each one at a time, and then you reconstruct this back in a computer, and then you get essentially a complete picture of what you were trying to map. And this is just the analogy that we used here. Just as kind of a side note as well, is that it's the original method described these 25 MZ windows, and we had 32 of those to cover the whole space. But relatively early on, we realized that the distribution of signal across the mass range is not even. So it's in this range from kind of 400 up to 800, we have a lot more peptides than in the range from 800 to 1200. So for the last few years, we've also been using more windows, but of narrower width, and also we vary this width across the mass range. So the method that we tend to use in the lab at the moment by default is this 64 variable window method, where in the most dense parts of the mass range, the windows are down to around 6 or 7 MZ, and up here in the last window where there's not really a lot of peptides, then we can have 90 M over Z windows, and this allows you to kind of distribute the signal more evenly per precursor isolation window, and hopefully distribute the signal and make it a little bit easier to interpret the peptides down here, which are in this most busy space. It's also important to say that there's many different instrument platforms where this can be done. So we've mostly in the past been using quadrupole time of flight layout, because at the time that this was started, there actually wasn't a quadrupole Orbitrap instrument available. So we've been mostly using QTOFs, and now more recently a little bit quadrupole Orbitrap layouts, either in the form of a kind of Q-exactive or fusion geometry, and this also works very well. And I think you might also hear, so the kind of main constraint seems to be that you would need some kind of quadrupole for mass selection for these large precursor isolation windows, and it's good to have a high resolution or medium resolution mass analyzer in the back, although I think you might also hear some developments on low resolution DIA from Mike McCoss later in the week. But just to say that this is certainly these are the most popular implementations, I would say. Okay, so now we know roughly what the dimensionality of the data is. We also need to know something about how to extract the data. So I said that our problem is essentially that we have these mixture spectra. And so here's now a depiction of these three peptides, and these are all falling in one swath isolation window. So let's say this is a 25M over Z window from like 500 to 525, and here's these three precursors, the red and purple and yellow one. And the question is, what happens in the experiment is we fragment these all together. Well, we get a mixture spectrum which looks like this. So we have fragmentines from purple, red, and yellow, but of course we don't know the colors. So the question is, like, can we actually, people sometimes ask, can we interpret this data in the kind of classical DDA way? And the answer is no. I mean, if you ran this this type of spectrum into Mascot, for example, this probably wouldn't work very well. You might identify kind of one of these peptides, but it's probably not possible to directly interpret these spectra by standard database sequencing approaches, database searching approaches, or at least not without some extra effort to do deconvolution and so on. So this is basically the problem statement. This is another view of that in essentially real data. So here's another swath MS2 spectrum, this time from 600 to 625. So these are all fragmentines from a whole bunch of different peptides, whatever was in this precursor isolation window at that particular time. And this is, again, another spectrum that you would struggle to interpret in a classical way with a database search engine. But let's imagine that we wanted to go after a particular peptide that we knew might already be in there. So take this particular peptide here, and what I've done now is to highlight the kind of the major fragmentines of this peptides in this spectrum, which I happen to know is in there. And the point I want to make here is that essentially these are the major fragmentines, which you can just about see down in the grass here in this spectrum. These are the major fragmentines for this peptide. And in this swath MS2 spectrum, they're not at all the major fragmentines for this spectrum, right? So trying to get this peptide out of this spectrum is going to be difficult in the classical way. However, there might be another way to do this. What if you had some prior knowledge about this peptide? You might be able to selectively extract this signal out of the data, and this is the basis for the data analysis method that we most commonly use. So this is what we in the past have called kind of targeted analysis workflow, or also is referred to as peptide-centric analysis, because you're taking a particular peptide and you're going to look for that peptide in the data. So let's say we have now some swath data where we acquired this spectra in this precursor isolation window. Here's again one of these very complex looking MS2 spectra that it's hard to interpret in the classical way. But what if we say we're actually going to try and find this peptide? Well, here's the fragmentines projected in a mirror plot for this particular peptide. And we can zoom in and say, is there evidence for any of these fragmentines in this very complex spectrum? And you see here for example, for this Y5 fragmentine, which is supposed to be at this position, we can see this isotopic envelope. So this is kind of a good sign. Now what we can do is look at for all the other fragmentines as well, and then we can do extracted ion chromatograms. So if we draw a little box around this and we extract the signal as a function of chromatographic retention time, and then we do that for each of these fragmentines, and then we plot these, then we can see a signal group here. And those of you who are kind of familiar with doing SRM or PRM type analysis will be familiar with this type of picture. So now we're in the situation where we have kind of pseudo SRM data, right? So it's fragmentines, the intensity of fragmentines plotted as a function of chromatographic time. And so this looks like it could be our peptide. And we have, of course, and this is, as Rudy mentioned, this is a large focus of the course, is how do you computationally get this data out of this very complex, how do you get the information out of this very complex data? And so I refer to this mProfit algorithm from Lucas Reiter in 2011, where we had systematic scoring systems for SRM data. And the pipeline that we use for SWATH data is essentially an adaptation of this. So we score various different things, like do all of these fragmentines coalute in the retention time domain? Do the fragmentine relative intensities correspond to what we know about this peptide already? So that in terms of utilizing this prior knowledge, are all these fragmentines, do they have a similar peak shape? If you have a reference, like a heavy reference, do they correlate with the reference and so on? And there's many other scores that can be looked at to determine whether this signal group is indeed the peptide that you actually started out looking for. And this is going to come up. This is going to be the topic of a couple of lectures tomorrow. Yeah. So if you like this three-dimensional plot way of seeing things, then this is a depiction of this here. So here's again this SWATH MS2 spectra. So this is the fragmentine spectra from one precursor isolation window, let's say 500 to 525, and these are all the MS2 spectra stacked up as a function of time. And now you can imagine taking some prior knowledge about this peptide that we're interested in and projecting this on. So let's say this blue spectrum, which is shown behind, is what we already know about this peptide from some prior DDA analysis, and we know that these are the various fragmentines that occur for this peptide, and now we project this essentially onto the SWATH data. So we can say we're going to selectively look for these fragmentines, and what you see is that we see a set of fragmentines that coalute, and if we project this, if we collapse this back down onto just the retention versus intensity dimension, here we have this signal group, which kind of again looks like SRM data. And this is the data that we actually are going to score, but it's important to remember that there's the full scan spectra are underlying this extracted data, and we can also score this kind of data as well. And so the software that we typically use that was developed in our group for doing this type of analysis is called OpenSwab, and we'll also be looking at Skyline during the week and also discussing various other packages, but this is the one that we mostly use in our lab. So this is open source C++, and it's compatible with open formats and so on. But essentially, the inputs that you need is basically a spectral library. So this is the prior knowledge about how these peptides are going to chromatograph and how they're going to fragment. Then you need some DIA or SWATH data, and essentially you can well, first we do a little bit of retention time alignment, but essentially what we're going to do is use the spectral library to extract these fragmentine, these extracted ion chromatograms at the fragmentine level, and we're going to say, okay, how do these signals look like with respect to what we expect them to look like, and we can score them in some way, and then we can have some kind of statistical model that says which is the correct signal group and what's the false discovery rate and so on. So this is all going to be discussed more tomorrow. I just want to give you a preview of that, and this is also one of the tutorials is focused on is the use of this pipeline. Okay, so let's go back to, again, thinking about what the data actually looks like and make some comparisons to more targeted methods. I think this can be instructive. So let's compare SRM to DIA data. So in SRM, we have basically this triple quadrupole mass spectrometer, and where we have some complex mixture of peptides being sprayed into the mass spectrometer, and we have a Q1 where the mass, the isolation width is set to typically 1 or 0.7, something on that order. And let's say that we're trying to measure this green peptide, right? So we isolate that precursor, but sometimes you have another, this red one is at a similar nominal mass, so this red one is coming along for the ride. And then this peptide gets fragmented by collision-induced dissociation, both the red and the green one. And then the trick with SRM is that you get selectivity by just filtering out one of these fragmentines at a time. So we try to acquire data on this particular green fragmentines. And now this is a kind of a pseudo MS plot of the SRM data, right? So these are the 3 fragmentines that we're trying to monitor. These 2 have nice clean signals, but unfortunately, this red one, one of these red fragmentines happens to have a similar mass to this green fragmentine, and so we get an interference in this case. Now let's consider what's the situation in DIA or SWATH mode. So we have the same set of peptides being sprayed into the instrument here. And now instead of a triple quad, what we have is we replace the third quadrupole with a high resolution mass analyzer. In this case, is time of flight, but it could be Orbitrap or something else. And now what we're doing in DIA mode is, in order to get this continuous sampling of the precursor isolation space, what we're forced to do is to open the width of this precursor isolation window. So up here in SRM, we had unit mass resolution, we had unit mass precursor isolation, and down here we've opened this up to 25 mass units. And now what you can see is what passes through this first quadrupole is more complex than up here. So here we had the red and the green guy, and now we have the purple and blue ones coming along for the right as well. So these all get co-fragmented, and now we have purple and red and green and blue fragments, and these are all analyzed by the TOF mass analyzer. The thing is here is that we have much higher resolution with this TOF mass analyzer than we do with this quadrupole. So what that allows us to do is to say, okay, here's these same 3 green fragmentines that we're interested in. And what we essentially can do is now do an extracted ion chromatogram for each of these fragmentines. And notice that the width of this box is rather narrower than this Q3 isolation width here. So we can, because we now have high resolution spectra here, we can buy some sensitivity back here. So we lose sensitivity in the Q1 by letting more things through with comparison to SRM, but we buy some selectivity is what I'm trying to say. So we buy back some selectivity by having a narrow extraction window here. And now these green guys are all clean, so this red interference is no longer an interference as it was in SRM because of this narrow isolation window. But of course the problem is that we don't just have all of these, we don't just have these 3 green fragments, we have everything else. We have all of the fragmentines from these 4 different peptides. So this is this very complex mixture MS2 spectra that I've been talking about. Which is difficult to interpret in classical ways, but if we just try to selectively extract out the information that we want, so if we know in advance that these are the best 3 fragmentines, then we can pull that information out without worrying about what else is in the MS2 spectrum. It's also possible that some of these fragmentines, so for example, if we look at this green fragmentine here, then there's an interference. It's hard to see, but there's a little blue one here beside that. So, I mean, that's not what we want. We wouldn't want to use that fragmentine for extraction because it's going to be interfered from this signal. But the benefit of having this high resolution full scan spectra behind is that you can just pick some other fragmentines to monitor if this one is interfered with. So now let's look at the comparison between PRM and DIA. So up here we had SRM, now I'm going to switch this to PRM. So what's the difference with PRM? Now in PRM we still have this narrow Q1 isolation, 1 mass unit wide, but instead of the 3rd quadrupole, we again have some high resolution mass analyzer. So now we have both selectivity, we have a selective Q1, but we also have a selective extracted ion chromatogram because we have high resolution data here. So now we have kind of the best of both worlds from a selectivity perspective. So this is the kind of the most selectivity you can get of the 3 methods. But again, with PRM, you have the problem that we're not covering the whole mass range. So it's very high selectivity, but it's not comprehensive. And the trade-off here is that in DIA mode, we have lower selectivity, but we cover the whole mass range by opening up this Q1 precursor isolation window. And so this sort of concept of doing kind of targeted data extraction was published in 2012, this paper from Ludovic Gillet, who will be here tomorrow. And the idea, the high-level concept there is if we take this kind of complex DIA data, which is very complex and difficult to interpret, but we build in this prior knowledge, we say we know something about the peptides that are likely to be in this sample, and we're going to try and selectively extract that information with reference to the information that we already know about these peptides. Then we hope we can get back to something that kind of looks like the quality of SRM data. And that's the point, that was the point of this paper, and this is kind of the concept that we've been trying to push since then. So it's important to realize that I said we need to have this prior knowledge about these peptides, and this comes in the form of a spectral library or sort of peptide query parameters, we also sometimes call this. And this is basically just a table which says, for this protein, you know, we've previously observed this particular peptide. We know what the precursor mass is and what the charge state is. We know the likely set. We know what the most intense fragmentines are. So this is the set of fragmentines. This is the set of their masses. We also know something about the intensities of these fragmentines, right? So this Y8 one is the most intense, and we know something about the rank of their intensities, so this is useful prior knowledge. And we also know something about the normalized so the retention time, their chromatographic behavior in some normalized space, which I'll talk about a bit later on. But essentially, this is the set of information that we're interested in having when we want to go and extract selectively in this kind of targeted analysis paradigm or this peptide-centric analysis paradigm. This is the set of information that we need to know before we go to the DIA data and try to extract out information from these peptides. So what might some of the advantages be of DIA over more targeted, or well, let's say other methods? So this is now a figure again from this earlier paper from Ludovic, 2012, and this is supposed to try and depict how we think, or at least how we thought at that time, these different methods compared to each other. So this is a dilution series of this peptide into a yeast background, and we measured it either by using DDA MS1 quantification, SWATH, SRM, and what's also plotted here is the mascot score in the DDA. So the sort of circles and the squares are coming from the one method. And what's plotted on the x-axis is this is a dilution series of this peptide, right, going from 100 femtomoles down into the attomole range for this single peptide. And what you see here is plotted the response for any of the methods, and for the purpose of the DDA, this is just a mascot score. That's why it's kind of flat like this. And what I'm trying to emphasize here is what we think that the sort of, roughly speaking, the order of the sensitivities of these methods was at the time, but I think it's sort of, based on our experience, it tends to follow this pattern more or less. So this is the limit for identification in DDA, in mascot, right? So this is the last concentration where this peptide was actually identified in the DDA analysis. This is the limit for MS1 based quantification, so that's square actually, and this is limit of detection. You can see it's kind of not really linear, but this and the black triangles is the dilution curve for SWATH. So we go down basically into the attomole range for this particular peptide, and then the X is SRM. So this is still in most of the time the more sensitive way to do things. And this is roughly, people often ask, well, what's the comparisons in terms of sensitivity between these methods, and this is roughly still what we see at the moment, that SWATH is pretty good, but it's still not quite as good as more targeted methods like SRM or PRM. Because we developed this in the context of being mostly a targeted proteomics group at the time where this was developed, we tended to make a lot of comparisons with SRM. So this is just another example of that from Yansheng Lu. And here he's trying to measure formally glycosylated peptides in plasma. So this is using a glycopeptide enrichment strategy to kind of dig deeper into the plasma proteome. And this is a set of basically 37 peptides, which he could quantify by SRM. And this is the same data for SWATH. And what you see is that it basically looks the same, except there is a slightly, we have only 33 peptides quantified instead of 37. So this again reflects the kind of slight gap in the sensitivity of SRM and SWATH, but it's qualitatively very, very, very similar. Also, from a kind of precision perspective, we get a similar level of precision as measured by these CVs of these peptides. So this is just sort of roughly to give you an idea and position where this type of data is with respect to other types of data. Another example of this is also, again, from Ludovic's initial paper. There they looked at this a yeast, kind of a classical experiment in yeast biology called the dioxic shift, where you start feeding it glucose, and then it uses all the glucose and it turns to ethanol as a carbon source. And so you get a kind of a metabolic shift as part of the experiment. And this was a system that was worked on in our lab a lot for SRM analysis. So this is a whole bunch of proteins in central carbon metabolism that were quantified by SRM. And now if we do the same measurement by SWATH, we get essentially the same picture, right? So this is just trying to say that we can retrieve the same information as you can from SRM with almost the same sensitivity, but not quite. So again, in this case, it was like 43 of these peptides detected by SWATH out of 45 detected by SRM. Okay, so what about kind of added value over other methods like SRM? Well, so this is now a picture of this peptide, the extracted ion chromatograms, and we're monitoring this peptide at 522. This is the precursor, and now we're monitoring these three fragment ions, right? So this is actually DIA data, but it sort of simulates an SRM experiment, what an SRM experiment might look like. We're looking at these like Y6, Y7, Y8 fragment ions, and now we see we have kind of two peaks which look fairly reasonable. So which one is the correct one? And the problem is that in SRM, you would have the issue that you'd have to go probably back and do remeasurements to figure out, with added transitions, to figure out which is the correct peptide. But in DIA mode, or in fact, also in PRM mode, you would have access to the full scan high resolution data. And so you can just say, I'm going to go and I'm going to add another 3 fragmentines. So now we can see with these two peaks, if we add these Y5, Y4 and B4 fragmentines, then we get a nice signal on one of these 2 peaks, and in the other one, these added fragmentines are essentially flat, right? So we have the possibility to go back and look in the data for additional fragmentines with comparison to an SRM type of measurement. And this idea of kind of iterative mining of the data will probably come up again. And what we would sort of argue here is that the DIA data is complete in a way that as either SRM data or DDA data is not. And this makes it kind of suitable for kind of data reanalysis potentially. Okay, so what about interferences? So here's a peptide that we are interested in measuring, and this is now in N15 yeast labeling. So this is the light peptide, this is the heavy peptide, and we're monitoring the same set of transitions for the light and heavy peptides. And you can see that one of these transitions has a kind of obvious interference in the heavy channels. So this is again the case in SRM where you would say, oh, well, too bad, I have to go back and do remeasurements if I think that this is a big problem. But whereas in DIA data, you can just say, well, I don't want this fragmentine anymore. I'm going to pick instead a new one which is not interfered with in either of these channels. So this is just trying to emphasize the idea, the completeness of the data, and the idea that you can kind of dynamically decide how you want to extract this data. Similarly, you can do this at a kind of a higher level. So this is, again, this dioxic shift experiment looking at this kind of central carbon metabolism TCA pathway. And this was the experiment that they measured by SRM, and they also measured it by SWATH. And now the biologists said, oh, well, we don't want to just know about TCA cycle. We also want to know about some other pathway that could have been affected by this perturbation. And in this case, it was an electron transport chain. So in the SRM example, you're out of luck because you've already done the measurement. But in the DIA data, we can just go back and say, okay, we're going to try and re-extract the information for all of these ATP or these electron transport chain complexes. And the idea, this kind of goes also in the direction of kind of iterative data mining, that you can, the data should be there within the constraints of the measurement, and you can go back and look at the data over and over. And this is kind of what I try to, so Rudy already showed this picture, but essentially what I would emphasize is this kind of, we're trying to go for data completeness here, both from the respect of the matrix, but also in terms of whether the data can be reanalyzed. Okay. So the summary here is essentially that DIA, this is the same list I showed at the start, and maybe this makes a little bit more sense now. So the summary is that DIA has this fixed duty cycle. It's entirely deterministic, right? So this we said is kind of unlike DDA, but more like SRM or PRM, and this is what we think leads to a significant degree to the completeness of the data and the ability to do robust quantification. It's also somewhat untargeted and unbiased data acquisition. So this is kind of unlike SRM or PRM, where you're restricted in the set of things that you want to monitor, but more like DDA. We have comprehensive sampling of the precursor space with MS2 spectra. This is a characteristic feature of DIA, but this is usually coming at the, this is usually achieved by looking at these wide precursor isolation windows. And this is usually coming at the cost of having very complex mixture MS2 spectra that are much more difficult to interpret than classical DDA-based MS2 spectra. And so this kind of targeted analysis paradigm or peptide-centric analysis paradigm based on prior knowledge is what has really helped to make DIA feasible, at least in our hands. Other people are doing this different ways, but we still very strongly take this kind of targeted or peptide-centric approach to the data analysis. We think that the data are roughly SRM-like in terms of their quantitative characteristics, or at least what I say is approaching SRM. It's often not quite as good, but it's getting there. In terms of the data completeness and linear dynamic range and LOD, LOQ, and so on, we're pretty close to SRM. And to me, the big benefit of DIA data is its sort of flexibility. So you can do classical targeted proteomics analysis, kind of SRM-like analysis of the DIA data, where you say that there's just 50 proteins that you're interested in, and you try to extract the MS2 level quantitative information for those 50 proteins, or you take the position that you want to try and extract quantitative information for all the peptides and proteins that you can, and then you're in a more sort of discovery-like mode. Okay, so maybe a little bit of outlook. See how I'm doing here. Pretty good. And this is what I just mentioned. I think this kind of theme is going to come up over the course of the course. That there's different ways to analyze the data. You can do it in a much more focused way, where you only looked at the peptides that you're interested in, or maybe the peptides that you actually know are already in the sample, or you can do the data analysis in a way where you try to find everything that you can. And this is kind of more in the SRM tradition, and this is kind of more in the DDA tradition, and there are things in between too, right? So you might have some kind of weak hypothesis. But I also, this arrow is to remind me that this type of analysis can probably be done iteratively because of the completeness of the data, if that's what you're interested in. But the important thing to know, and this is going to come up over and over again across the week, and it came up a little bit already in Rudy's talk, that as you ask more questions of the data, the more questions you ask of the data, the bigger the statistical burden is in terms of multiple testing. So you lose sensitivity by asking more questions. So it's actually probably important to keep in mind that you should only ask the number of questions of the data that you need to in order to avoid this kind of burden of multiple testing correction. But as I said, what I think is one of the key sort of advantages of this type of data is that you're flexible to operate in basically any of either of these paradigms or anywhere in between. I just put this up because I think it's interesting. We've been kind of hammering away on this type of methods along with other groups like the McCoss group and a few others for quite a few years. And it seems to me in the last 1 or 2 years that there's also some other groups which might have been more DDA traditionally focused who are considering the idea of using DIA. And I just show one example here from Jesper Olsen's lab, where they published this paper about evaluating the performance in the HFX. And the paper is about different things, but the last section is kind of a comparison between DDA mode and DIA mode on this HFX instrument using either MaxQuant or Spectral for the data analysis. And their conclusion, I think, is that the DIA analysis was a little bit better. This is what's shown in this histogram here, the set of proteins they detected. So the blue is the single-shot DDA, the red is the single-shot DIA, and I think the light blue is basically a fractionation of the same sample. And their conclusion was that DIA is very kind of promising in this respect, and so it's nice that the kind of broader community is starting to come on board with this a little bit. We hope. We haven't been operating for no reason. It's also interesting, I think, what's going to happen to talk about what might happen in the future. So as instrument scan speed continues to increase, you could imagine that you would end up with a method where the DI precursor isolation shrinks to basically unit resolution, or equivalently, the DDA sampling becomes so dense that you have essentially MS2 sampling over the whole MZ and chromatographic space, or you could imagine it like PRM that can comprehensively sample the whole precursor space, right? These are all basically the same thing, and this might eventually happen. And you end up with like a kind of super method that just samples very densely the precursor space with narrow precursor isolation windows and high resolution full scan MS2 and so on. And then I think the interesting question is going to become what do we actually do with the data? So you could analyze the data in this so-called kind of spectrum centric way. So you start from each individual spectrum and you search it using Sequester or Mascot or something. Or you can analyze the data in the way that I've described by looking for particular peptides and trying to selectively extract them out of the data. And I suspect this is the one that's going to be better for quantification, although we might see some, and we have already seen some kind of hybrids of these kinds of methods that might be very interesting going forward. Okay, so that's basically what I wanted to say. I think I even am a little bit ahead of time, so if there are any questions, please let me know. Otherwise, okay, it's a little bit too fast. I actually can't see our next speaker in the room, which is a small problem. Could you check if Olga is here, Amir, maybe? Yeah. Oh, I'm sorry. Yes.
Regarding this multiple testing burden that you mentioned, would you then advise not to use like the full proteome to query your data against, but rather just the observed proteome, which we saw in the talk before?
Yes, so that's going to come up a bit in the lecture in the afternoon. I think there's no point in querying for things that aren't detectable in the data. That can only hurt you, right? That's if you look for a peptide that you somehow magically know isn't in the sample, then that peptide, that channel that you're extracting from, all you can do is accumulate false signals in there, right? You can't. So, and this will eventually change the score threshold at which you achieve a particular fixed FDR. So you will lose sensitivity that way. And you may even say that for even for things that are detectable in the data, but you're not interested in them, you could also leave those out potentially too, right? So yeah, I mean, the general trend, the general case is the more questions you ask, the less sort of sensitivity you have. And if you can be more focused, then I think that's, from a statistical perspective, useful. And that also goes for not just detecting the peptides, but also doing like differential expression analysis as well. You don't necessarily want to test for everything that you're not interested in, because this is also going to increase multiple testing burden. Other questions? Yeah.
So you mentioned completeness and the linear range. That was for the triple TOFs, but is it the same on Orbitrap instruments?
Yeah, that's a good question. So, so I think I'm going to duck that question and leave it. We're going to have some talks later in the week from Ludovic Gillet, who's going to talk specifically about some differences between TOF and Orbitrap in this perspective. And we're also going to have some talks from Mike McCoss, who's much more experienced in doing Orbitrap DIA. I think maybe I'll do a one-line question, which I think that our feeling is that probably the latest generation Orbitraps are potentially more sensitive than the QTOFs, but I think that's because potentially, well, Thermo has been putting a lot of effort into development and releasing a lot of new platforms, and for example, the triple TOF platforms that we've been working on have not been incremented so often. But yeah, I think this will come up later.